Awesome. Thank you, Dawn, and thank you everyone for having me. This is my first Woodland Conference, so I'm really happy to be here. So today, I haven't been as involved in the Town of Truro Urban Deer Management Project. I'm still getting caught up. I've only been the manager of the team for the last five or 6 weeks now, but I've been on and off on the team since 2017. So my involvement in that project specifically wasn't very significant, but my background is in wildlife disease surveillance. And so this research that I'll be talking about today is, it doesn't impact deer so much, but deer are the ones that spread the disease, and it impacts our mainland moose and our Cape Breton moose. So that'll be the focus today. There we go. Okay. So like I said, I'm going to be talking about 2 different projects today where we were sampling deer. So COVID-19 surveillance will be the first one I'm talking about. And then the second one will be brainworm. So COVID-19 surveillance was in partnership with a lab in Saskatchewan, and then brainworm surveillance is in partnership with Acadia University. And all of the deer heads that we used for this research were sourced either they were donated by hunters in the fall, or the department does an annual winter roadkill survey. So we're already looking at deer body condition in the winter and fecundity, and then we were able to just also use the heads for additional disease research. And this research would not have been possible without our funding sources, so we have a really close relationship with Acadia University, and there's something called the Habitat Conservation Fund. So if anyone isn't familiar with this fund, for hunters and trappers, if they want to buy their licenses each year, they have to buy what's called a habitat stamp or a base stamp. And the money from those stamps goes into a research fund. So anyone can be applying to this fund in terms of research, whether it's about wildlife or habitats. And these applications go in their 1-year research grants. And so we've done this research for 4 years, and each year we've gotten Habitat Conservation Fund money, as well as a federal NSERC grant, which is through Dave Scheller specifically. So, as I mentioned, if you're interested in looking into the Habitat Conservation Fund, anyone who receives these grants each year, the award winners are posted online on the website every year, so you can actually see where the money is going, who receives the money, how much money goes to each project. So we'll start with COVID-19 surveillance. So we, as a department, were approached by Saskatchewan, who after the beginning of the pandemic, this was around 2021, the Prairie and Northern Wildlife Research Center in Saskatoon contacted various jurisdictions and provinces to see if we could look at COVID-19 surveillance in wildlife, and more specifically cervids. So their objective was to evaluate whether wild deer and relatives have been exposed to or infected with COVID. And the methods were similar to how we were getting tested for COVID. So it was a nose swab and a throat swab, and then those swabs get sent off for genetic screening. So as I said, the project started back in 2021, so this is just kind of a summary of our results. So back in 2021 were our early days. We had about 20 deer heads that we were able to swab, and they all came back negative, so we didn't have any COVID positive cases there. Then in 2022, we had the opportunity to swab 135 deer heads, and 9 of them came back positive for COVID, and they were able to genetic screen them to the specific variant. And then again in 2023, we swabbed 129 deer heads, and 6 of them came back positive for COVID. And then in 2024 is where we kind of wrapped up our screening for now. Those samples just got sent off in early 2025. And they're still being processed, so we don't have those results to date. And if you're wondering where these COVID deer are in the environment, so we have several positive cases out of the Truro area, and then we also had a couple positive cases up in Cape Breton as well. So the goal of the surveillance was to use data to help assess potential transmission pathways and risk for establishment of coronaviruses within populations, and evaluating potential risks of spillover and spillback at the human-animal interface. So Saskatchewan is compiling all the data they've received from various provinces for the last 4 years, and they're doing a nationwide publication. So I'm hoping the discussion portion of that publication is really going to get into this and what the risks are. And as I mentioned, 2024 is where we kind of kind of wrapped up, so we have ceased sampling to date, and like I said, we're working with Saskatchewan on that publication. So that's kind of where we're at for COVID-19. We weren't doing any of the genetic screening, we were just strictly kind of doing the swabbing and sending them off. But then the brainworm surveillance is kind of where we've done more of the work out of our lab in Kenville. So brainworm, or it's also called P. tenuis, is generally benign in white-tailed deer, but it's often fatal in moose, so this is why we were looking into it. And prevalence and distribution of brainworm have not been evaluated in Nova Scotia, or at least published in the literature since the 1980s. So we don't have any recent data that we can look at. And up-to-date information could help with moose management both in Cape Breton and on the mainland, and I'll get into that. So I'm going to get into the life cycle of brainworm because it'll help you understand why it impacts moose differently than it does deer. So the adult worms live on the top of deer brain, and so it's they're nematodes, they're roundworms, so you need a male worm and a female worm to be able to reproduce and have that deer actually shed eggs into the environment. So they live up in the skull cap there. These larvae, once they travel, so they go through the blood and into the lungs of these deer, and then the deer cough up the larvae and then they swallow them. Once they've swallowed them, they go through the stomach, through the digestive tract, and they get pooped out into the environment. Now, we know slugs and snails like to feed on larvae and nutrients-rich things in the environment, so the slugs and snails are what we call the intermediate host in this life cycle. So the slugs and snails will actually feed on the infected deer poop and pick up these larvae. Now, we know deer as well as moose like to graze, eat browse, so they're actually accidentally picking up these infected slugs and snails in the environment. Now, once the deer, I'm going to focus on deer, once the deer have the infected slugs and snails in their stomachs, these larvae now migrate through the stomach tissue, through the blood, and to the spine, and then they travel up the spine and then up the back of the brain with causing as little damage as possible to get to the top of the brain, which is where they live. So these worms specifically are programmed to do this in deer without causing damage for the most part, because you want your definitive host or your main host to live healthy so that you can continue to survive and reproduce. So I'll bring it back to moose here. The issue with moose is that they are called the dead-end host. So moose are still picking up these infected slugs and snails in the environment. However, these larvae are going up the spine and they're not able to get to the part of the brain where they're supposed to go without causing damage. A moose is much larger than a deer. So what ends up happening is these worms are getting lost in the brain of the moose and they're burrowing tunnels through the brain tissue, which causes immediate neurological damage, and it's irreversible. It's not something you can really just give them an anti-parasitic and it will fix. So if anyone's ever seen videos or unfortunately seen in person, what you kind of see is those zombie moose where there's been some videos circling online where the moose, their head will tilt. They have hair loss on their head because they're rubbing their head. They'll walk in circles. They have next to no fear of people at that point because they're just kind of wandering. And the dead end part of that is it's very unlikely for larvae to get to the top part of the moose brain in order to reproduce and have the moose actually shed eggs into the environment. So the moose end up dying well before that happens. So the moose aren't shedding these through the environment, but the deer are. So this research is primarily led by our Acadia Work Skills students. So the Kentville office for DNR is really lucky to have Acadia, which is in Wolfville, just a couple of towns over, so close by, where we have this relationship and this funding where we can hire Acadia students to work part-time out of our lab. So Willow Bennett on the left there was our first honors student who started this research, and then we've had a few other research students, Mathieu and Jacob, and then Gareth here is the other one in the photo. He was our last honors student for the full 4 years of surveillance, and we're all working together now to kind of get this research published. So this is kind of a summary of the years and the deer that were tested. So in 2021 is when we were starting up. So these numbers in terms of amounts are going to be similar to COVID-19 surveillance but different. And that is because for the deer heads that were submitted for this research, the skull had to be... Hello, okay. The skull had to be intact in order to properly look for the worms. So unfortunately, if it was a hunter harvested deer and it was a headshot, or if it was a vehicle collision and the skull was quite shattered, we couldn't properly survey for brainworm, but we could still swab for COVID. So we wanted to make sure we were using as much as possible. So that's why the sample numbers, we were still using deer heads for both projects, but the numbers are a little different. So in 2022 and 2023 is where we were really steadily sampling. So we had over 100 deer heads for both of those years. And then in 2024 is where we were winding down with only 26 heads. So this is what we are doing in the lab. We have a vise that is bolted to the wall with teeth, and you basically put the deer head in the vise and crank it in so it doesn't move. And then you saw the skull cap off, because where those worms live are directly underneath the skull cap within the tissue, and they're visible to the naked eye. So you can see they're kind of a very dark worm on a nice light pink background, so they're relatively easy to see. And what we do is we remove them and we put them in a petri dish and we look at them under a dissecting scope, and we try and count them as well. So we know if they have at least one, we know that deer was infected, but we want to know how bad these infections are. So we're able to count the worms under the microscope as well. So if the deer only has a single worm, we know that deer wasn't actually shedding anything into the environment because the worms couldn't reproduce. But if it has two or more, you know that deer is likely pooping larvae into the environment. So this is the map of the 4 years worth of data that we looked at. So the green is negative, which is good. We really don't want brainworm in the environment, especially where we find mainland moose and Cape Breton moose. And you can see it's pretty evenly distributed across the board in terms of where we sampled, as well as where you find brainworm. It's unfortunately kind of everywhere. The only plus I would say is we know we have a pocket of mainland moose up this way at the isthmus, and it's all green there. So perhaps there's something going on up that way where we don't necessarily have brainworm there, where we know moose they come and go from New Brunswick. So that's a plus, but in terms of modeling, we're still working on some results as to what this could tell us. And so this is again another table kind of reviewing the results. So you can see by year prevalence, which is the percent of our sampled population that was infected with brainworm. So in our first year, because we didn't have many samples, we had a prevalence of about 25% of our samples were positive. In 2022 and 2023, it was about 50% both years, and then 2024, again, with fewer samples, was only 35%. Then we were also looking at deer density in relation to brainworm infection. So the first one here is prevalence. So again, percent of your host population infected. So as you can see, as deer density goes up, which means more deer per kilometer squared, so when you have more deer in smaller areas, we also have your rate of brainworm infection goes up as well. So it makes sense, you have a lot of deer in a given area, they're all feeding on the same things, their likelihood of having brainworm is higher. And then we found the same thing with intensity as well. So intensity is the number of worms per infected individual. So you can see here again, as deer density is going up, the number of worms, so adult worms in these deer, are also going up. So we're seeing higher rates of infection as well. And so I have a little video in terms of deer density here. So this was put together by Jason Ayers. I'm just going to get Chris to switch over. And so this is just going to show deer density from the 1980s up until today. So the purple and blue is kind of 0 to 2 to 3 deer per kilometer squared. And then once you get into the green, yellow, and red, you're getting into the 4 to 7 or 8 deer per kilometer squared. And so all this density data for deer is collected by Natural Resources. Each spring we do pellet group inventory surveys, and so that's where staff go out in the springtime and they walk 1 kilometer transect lines and they look on either side of them, and then any piles of deer poop that they see, they count. And they also look at hair pellets as well. Yeah, so you can see at the end there, you can see Truro density gets quite a bit higher, Yarmouth gets quite a bit higher. And the reason for that, just at the very, very end, is because we do have this urban deer problem, and we have these municipalities hiring private to actually do these surveys, these pellet group inventory surveys, within towns and on private land as well. So it's not that the deer population just exploded in the last 2 to 3 years, it's definitely going up. But it's because we've added additional data into those areas specifically. So it's still relatively accurate in terms of deer per kilometer squared. Okay, and this is my last slide here. So this is just looking at the prevalence that we found in the last 4 years and comparing it to some old literature in terms of Nova Scotia as well as nearby jurisdictions to see what our prevalence rates look like. So the last old data I could find was kind of from the 1980s, like I said, and they had said Nova Scotia had a brainworm prevalence rate of 65% of the deer in Nova Scotia, and our average overall was about 51%. So I was trying to figure out why ours seems lower, but we seem to have more issues. And what I found was the researchers back in the 80s, they didn't just look at deer heads for surveillance, they were actually collecting deer poop throughout the province, and they were looking for larvae in the deer poop, and then they could also look through the deer spines to find larvae as it was migrating. So they looked for brainworm in 3 different methods where we only did 1. So it's a really important factor here. So I don't want to say that our prevalence rates have just dropped. We just didn't do all 3 methods of surveillance. We didn't have the capabilities of doing that for this research. So I think that's important to note. And then again, I really couldn't find any up-to-date literature on brainworm for other jurisdictions. It's not that it's not there, it could just be buried in Excel sheets. So I'm going to do some digging in terms of contacting other jurisdictions to see if they have more up-to-date data. But again, everything seems to hover in kind of the low 60% range. And that's all I'd like to thank in terms of our partners, and I'm happy to take any questions.